All right, everybody, welcome back. In this video, we're going to look at Unit 7, Lesson 1, Exploration 2. We're going to continue our conversation about the process of meiosis. If you remember back to Exploration 1, we were looking at specifically chromosomes and the idea of homologous chromosomes, where we have chromosomes from each parent that are the same size and shape. They contain different details, but they contain the same types of information. And so how do we divide up those homologous chromosomes, and specifically, how do we then create the sex cells that we need in order to reproduce? So that's what we're going to look at here in Exploration 2, the process of meiosis. All right, now, meiosis, as a definition, is a form of nuclear division that divides one diploid cell into 4 haploid cells. Now, if you remember some vocabulary, diploid is where we have 2 complete copies of our DNA. In humans, that means 23 pairs or 46 total. And then that haploid description is what we have when there is half of the DNA that's supposed to be there, or just 23 total chromosomes, 1 of each. There are no pairs in those haploid cells. So in this process, we're going to start out with a diploid cell, and we're going to end up with 4 of these haploid cells that we call gametes. And then depending upon if they're male or female gametes is whether we're going to describe them as being sperm or egg. All right, so let's take a look at how this happens. First of all, we need to make sure we understand the difference between homologous chromosomes, which is what we talked about yesterday, and sister chromatids, which is what we had talked about when we looked at mitosis. If you remember, a chromatid is just a chromosome. It's the condensed version of a chromosome. So prior to this process beginning, each of us has 46 chromatids that can condense. And then during that S phase of interphase, all 46 of those chromosomes are going to duplicate and become sister chromatids. So if you look at this picture right here, this would be like chromosome 1 from your dad, chromosome 1 from your mom, and they are homologous because they are the same size and shape. After they replicate, they're still homologous, but now with each one of these, we have that sister chromatid idea that we had during mitosis, where we have 2 copies of that chromosome linked together. Now, in mitosis, we wouldn't necessarily care about the homologous chromosome piece, but in meiosis we do. And so let's go ahead and follow this down further to see what happens with these homologous chromosomes. All right. Meiosis is going to happen in 2 stages. We call them meiosis 1 and we call it meiosis 2. All right, so let's start off with meiosis 1. In meiosis 1, we are going to separate the homologous chromosomes, and we're going to end up with 2 haploid cells that each have that sister chromatid, that duplicated chromosome, a part of it. All right, so take a look at some of these images. This will kind of show you what happens. All right, so first of all, we have the prophase, which is very similar to prophase in mitosis. So prophase 1, we're creating those sister chromatids, we're condensing them together, we're ultimately going to be eliminating the nucleus so that they can move around. Okay, it's very, very similar to prophase 1. Again, I'd encourage you to go back through and click through all of these. Now, metaphase 1 is going to look similar to metaphase in mitosis with one key difference. Rather than having each of the sister chromatids line up individually along that center line of the cell, what we're going to have instead are the homologous pairs that are going to line up. So what you're going to end up with is you're going to have these 2 homologous pairs right next to each other. In fact, they're actually going to be right on top of each other. And as they line up, they're going to line up in these homologous pairs as opposed to as individual sister chromatids. Then we're going to move on to anaphase 1. And just like in mitosis, anaphase is where we are going to pull these 2 parts to opposite sides of the cell until we ultimately get to telophase, where we can reform the nucleus and we see these 2 new distinct cells forming. Now, the key thing here that I want you to see is that we don't have one copy of each chromosome. we have one homologous piece. So like in this picture, the white chromosome and the red chromosome are homologous. And we don't have one of each, but we have 2 copies of the red and 2 copies of the white. And again, this goes back to that idea of genetic diversity. The first way that genetic diversity is introduced in meiosis is through this process right here, where our homologous chromosomes are pulled to one side or the other. Remember, one of these chromosomes belong to mom, one of these chromosomes belong to dad, and the way that they separate is random every time a gamete is produced. All right, so let's go on to the next part. Let's look at what happens in meiosis 2. All right, so we've got our haploid gamete, and now what we need to do is split it up so that we don't have that copied sister chromatid together anymore. So in this stage, we're going to be separating sister chromatids, and what we're going to end up with is a new cell that only has half of the original information. So again, we look at this right here, and we can kind of follow both of these cells as we go through. Prophase 1, once again, they're going to condense, nucleus is going to go away. Now in metaphase 2, now those sister chromatids are going to individually align in the center of the cell, but remember, there's only half of them as what there would have been in a regular cell division. Anaphase 2, those sister chromatids are going to get pulled apart, and then telophase 2, they're going to reform in their new cells. Now what I want you to see here is that each one of these new cells only contains 2 chromatids. And they're different from each other. The chromatids down here are different from the chromatids up here. And if you scroll back up to our first image, we started out with 4 chromatids. And actually, technically, we started out with 8 because they copied themselves. So this kind of further illustrates that idea of being haploid. We only have 2 chromosomes here instead of 4. We do not have any sort of homologous partner with our chromatids. And that's on purpose because remember, the gamete's job is going to be to combine with another gamete that also only has half of the DNA that it needs. All right. The process of creating gametes in males is different than the process of creating gametes in females. And the major reason for this is because of the lifespan that's necessary for each of these gametes. All right. Male gametes are produced throughout the entire reproductive life of that organism, where female gametes are produced entirely before the organism is even born. In other words, males are producing sperm constantly throughout their entire lifetimes, but females have every single egg cell they're ever going to have by the time that they're actually born. And so because of that, these sperm cells are created to be much more temporary, much more fat, much easier to produce. They don't live very long. They're not very hardy. They're very easy to kill. Whereas egg cells are much larger. They contain much more cellular material. They have the same amount of DNA, but they have way more cytoplasm, way more organelles. They're way bigger. They're just able to handle living for the entire reproductive life of that female. All right, here's kind of a summary. This is a good image that kind of summarizes the difference between mitosis and meiosis. Remember, when you look at mitosis, we're going to start out with 1 copy of each chromosome. We're going to duplicate it to form these X-shaped structures, and then we're going to split them in half to where we end up with 2 new cells that each contain 1 copy of each chromosome. In meiosis, we're going to start out with the same set of DNA. We're going to copy it to create these sister chromatids, but then instead of trying to create 1 copy of each, what we want is actually half of the original DNA. So if you look down here at these are our haploid cells that are the result of 2 rounds of division that are going to try to create that genetic diversity that we're looking for. Again, mitosis is going to create cells that are genetically identical to the parent. Meiosis is trying to create cells that are as unique and diverse as possible. All right, here's kind of another good way to summarize this. I put this table together, so when you do yours, go ahead and compare it to this one if you're having trouble. But this is also a good way to kind of describe the differences between mitosis and meiosis. All right. So that takes us to the end of this exploration. Make sure you go back and rewatch anything you need to see again. But that's it.